Hello everybody and welcome back to my second lecture of this course on biomolecules. So, in the last lecture I have given a short introduction about the different biomolecules that are present in the living organisms, and I also have tried to show the importance to study the chemistry and the biology behind those molecules. So, and after that we have started with the first biomolecule that I picked up as DNA, nucleic acids, deoxyribose nucleic acids or RNA also, later on we will go on. So, what we have seen is that the background story of it, that with the help of the certain informations that were available during that time, that the DNA consists of the three major components, the one is phosphate, other is sugar, and another is of course the nucleobases. So, that is one piece of information. Second is of that number of equivalents of adenine is the same as that of thymine, and the number of guanine is equivalent to the number of cytosines. And with the help of the experiment, actual experiment, X-ray diffraction pattern of DNA that has been obtained by Rosalind Franklin and Maurice Wilkins, Watson and Crick finally came up with the structure of the DNA, that is the double helical structures and bDNA. So, and that structure is obviously unique in nature, unique in the sense that this is the first molecule, or this is one molecule, one biomolecule that is not a single entity. It is actually a combination of two molecules, and they are oriented in such a way that they have certain relations, and in this case the relation is hydrogen bonding and pi stacking interactions. So, that makes a single entity, but two different set of molecules. So, that is one uniqueness of DNA. Second is that I have already talked about, that it is the only molecule in the living cells, or even you cannot find out a similar molecule that can self replicate, that can make its own copies. So, because of this discovery, this has been a tremendous discovery during that time, because structure and functions of DNA was not been able to be explained until and unless the correct three-dimensional structure of DNA was elucidated. So, 1962 Watson and Crick along with Maurice Wilkins, they had received the Nobel Prize in the physiology and medicine. Rosalind Franklin was not included in the list because she died beforehand at a very young age. That is a quite a sad story. But on the happy end, on the happy note, Linus Pauling did not return empty handed also. So, in the same year 1962, Linus Pauling received his second Nobel Prize in peace. He was one of the key persons who were canvassing, who had a strong voice against the use of nuclear weapons, and for that in the same year as James Watson and Francis Crick and Maurice Wilkins received the Nobel Prize for DNA 1962, Linus Pauling received his second Nobel Prize for peace. So, that brings me to the structure of the DNA. So, this is the plain and paper model of DNA that shows the skeleton of the DNA, and I think I have explained yesterday also that you have this as a sugar unit, and the nucleobase is here, and then a phosphate unit, 3 sugar, nucleobase and phosphate. They are oriented in such a way that it forms a double helix, and I have mentioned that the sugar and the phosphate makes the skeleton. This is the backbone of the DNA, and then the flat aromatic nucleobases are perpendicular to the sugar or to the helix, that makes a perpendicular position. So, that the one nucleobase and then the second nucleobase are on top of each other. So, if I draw I will try to draw the structure. This is the deoxyribose, that means there is a OH here. Here there is no OH, is only proton and proton. This is 3' end. Here you have the nucleobase, that is the 1' end, 2', this is 4' and then comes the 5'. 5' has CH2 and then OH, O, that is connected to a phosphate. So, there is another sugar here and there is a OH. So, the 3' end, if I consider this is the end of this DNA, 3' end always has a OH group, free hydroxyl group, and the phosphate is, this is not a complete phosphate, this is O minus. So, it is basically a phosphate unit, PO4 minus unit. When it is deprotonated, it will be double minus. So, this and then again a phosphate O minus, double bond O, O minus. This is the terminal again. So, this is the 5' end of this. So, 3' end has a free hydroxyl group, 5' end has a free phosphate group. 3' free OH group, 5' end has free phosphate group. And now coming to the nucleobase. If I consider, I am just for the sake of simplicity, I am considering thymine for both cases, how it would look like. So, if you have the N, this will be perpendicular to it like this, with it is a 6-member ring. This part I am writing bold, so that it means that it faces towards you, and the other one is actually behind you or behind the board. So, this is the double bond methyl NH. So, it looks like this. Similarly, the other one is up the plane and this is the down the plane. Both are, I am writing both methyl. So, the front face with the double bond methyl is faces towards you, the other side is back. So, that makes a flat the plane perpendicular to the axis. And whenever you write a DNA, we usually write it in the direction of 5' to 3'. So, there is a style of writing the DNA. This is usually the practiced style from 5' to 3'. And of course, if you have the single stranded DNA, the 5' end has a phosphate. I am writing P circle as the phosphate PO4, and then 3' end has a free hydroxyl group. And these are very important actually. Later on we will see how the presence of the terminal groups actually makes a difference, and that we will see during replication process, DNA replication process. So, that is how it comes here. So, DNA is the genetic storage unit. Now, when it comes to RNA, that is the genetic storage in viruses, and the RNA also acts as a catalyst. DNA, of course, mostly natural DNA, that Watson exists as the Watson-Crick double helix structures. We call it double stranded DNA, dsDNA, and they have helical conformations which is of B form. We call it a B form. Alignment, of course, as I have talked, sugar and phosphates along the helical axis, nucleobase perpendicular to the helical axis. So, this is how the 3-dimensional structure of the DNA would look like. this is the DNA B form, which is right handed helix. So, here this way, and there are few features that are associated with the structure of this DNA. One is of course, that if you look one complete turn of the helix, that is starting from here to there up to here, this part, this distance, it is a complete turn of a helix. This is known as helix pitch, and that is about, it is written here, it is written here, it is about 34 angstrom, and per helix pitch there are approximately 10 base pairs or 10 nucleobases that are there, or exact is actually 10.5 bases, nucleobases are there per helix pitch. If you look here, one nucleobase and the second nucleobase down on top of each other, this distance between one nucleobase and the second nucleobase is, so distance between two nucleobases, two nucleobases means the neighboring nucleobases, is approximately 3.4 angstrom. And another important thing is the distance between the two helical moieties, the distance between two strands. One strand is represented by this pink, other one is by this purple one. This distance between the two DNA strands is known as the helix diameter. This is approximately 20 angstrom. So, that is how they are closed in. And because of this structure, now there are two important pockets that are formed within the DNA. This is the B form of the DNA that I am talking about, which is the most natural one. One is called the major groove, other one is called the minor groove. So, if you see, it forms here, it is a large pocket, little bit larger pocket in the helix. So, this is known as the major groove, relatively larger pocket. So, and these are very important because they are the positions where the other molecules bind and do chemistry or do biological transformations. Major groove is for the binding of the larger molecules, for example, proteins or other nucleic acids like RNA and so on. So, larger molecules would bind to the major groove because it has a relatively larger pocket. And another type is the minor groove here this part. This is a small pocket, low space, that is why this is called the minor groove. So, the minor groove is for binding with smaller molecules because the size is small and the space is small. So, minor groove basically binds with ions or smaller organic molecules for various kinds of biological transformations. So, these are very important aspects of a DNA double helix. So, now how is DNA present inside the cell? So, this is a drawing of a schematic presentation of a cellular nucleus, the inside nucleus and the other parts of the cell, and this is for the eukaryotic cell, and that means the higher organisms. Lower organisms are called prokaryotes and they do not have cell nucleus, most of them. So, this is a nucleolus and this part is basically the nucleus where DNA stays. All DNA, the genetic informations is stored inside the nucleus of the cells. And just outside the nucleus there is cytoplasm and endoplasmic reticulum, Golgi apparatus, mitochondria. So, inside the cellular nucleus DNA lives and most of the other biological molecules or biomolecules, they mostly stay in cytoplasmic area. And DNA is packed inside the nucleus as chromosome and they are packed in chromatin. The space is called chromatin. This is how a human chromosome looks like. So, now some statistics. If you look, so as I have said, the distance between two nucleobases is around 3.4 angstrom. Now, if you look at the human DNA total amount of chromosome. So, in every cell, every human cell, we have around 3 billion base pairs. So, 3 into 10 to the power 9 base pairs per cell. That makes, since DNA is double helix, that makes here total number of 6 into 10 to the power 9 bases per cell. Now, if you calculate the total distance between the nucleobases here, so that will come out as 6 into 10 to the power 9 multiplied by 3.4 angstrom. If you calculate, that is roughly equivalent to 1.8 meter or roughly equivalent to 6 feet. So, in our every cell, the length of a DNA is around 6 feet long. That is a large area, and all of it is stored or packed into the nucleus of the cell into chromatin. Now, an adult human being has around 100 trillion cells basically. So, now if you multiply that with 6 feet, a human body contains the total length of the DNA is equivalent to reaching the sun and back from there 600 times. That is a huge area that a DNA covers within a body. But our cells are small, so around 6 feet of length of DNA is packed inside the cell. And the longest DNA, it is composed of only 6 chromosomes. That is one type of Indian red deer, particular type of Indian red deer that has only 3 chromosomes in it. We have 46 chromosomes, that deer has only 3 chromosomes in it and around 3.3 billion nucleobases. Now, I will talk about something how the DNA are there inside the cells and how the multiplication happens. So, one interesting fact is that, of course, that many of you already know, that if you have I am writing the cell nucleus and we have around 46 chromosomes. Now, when cell division happens, this chromosome or this nucleus, so this is cell nucleus, 46 chromosomes are staying inside the nucleus. When cell division happens, this cell is divided into 2 new cells, and these are nucleus again, and all 46 of it goes there, all 46 of it goes here. That is the DNA replication. That is what Watson-Crick has found out, that had solved the puzzle, how the single cell is divided into 2 cells, both of them retaining the exact genetic informations. So, this is how through a process called replications DNA can unwind, which I will talk later. The all 46, it can make 2 different copies. The same 46 will be here, the same 46 chromosome will be here, and that is how our cells grows up. All of our cells, I am talking about eukaryotic cells now, higher organisms, all our cells has around 46 chromosomes, and this type of cell division is known as mitosis, and it is a diploid cell division. This is called diploid cells because it multiplies into the full 46 to 46, the process called mitosis cell divisions. So, the question is now that if all our cells are equal, all 46, then how all the human beings do not look like the same? If the genetic information is transferred fully to the offsprings, then why we all look different? And that is true for all other higher organisms, animals and all, so on. They look very different, we have different set of properties. So, the point is usually our cell divisions of course happens through a mitosis way, and from here further cell divisions happens, all mitosis, all 46, 46. There is one exception, that is start with the 46 and then it undergoes not mitosis, but meiosis cell divisions, where in the new cells it is not 46, it is 23, half of it will go haploid cell divisions. 23 means 22 plus 1, and here also 22 plus 1, haploid cell divisions, half cell divisions, and that happens in sperm cells or egg cells, father's sperm cells or the mother's egg cells. These are the only exceptions where cell division is not full, is not 46 to 46, cell division is haploid, 46 to 23 and 23. Now, so if this comes from a father, so same thing happens for mother, if this is the sperm cells, this is the egg cells from mother, and here also is 23 and 23 haploid cell divisions, and then the cells fertilized and would produce a mature cell with a single nucleus. They will be fused together, single nucleus. Now, the new cells will have together 46 chromosomes in the single cells, and that is how a completely new cell is developed. Now, which 23 will come out of from the mother, from the father, and which 23 will come from the mother is unpredictable, almost unpredictable. So, the new cell that has been generated is completely new, completely different than anything that exists. So, that is how the variation is created, that is how we all look different, that is how we all have different level of immunity, different strength and all these things. And that is a very good protection also that higher organisms have from the environment. The lower organisms such as virus and bacteria, most of them they have the same nucleic acid structures, they have the same genome structures. So, that is bad for them in the sense that if you can kill one cell through a chemical agent, you can kill the entire population, which is not true for higher organisms, because we have different immunity level, different strength. Now, the constituents of the nucleic acids. And DNA nucleic acids contains, as I have mentioned, 3 different segments. One is the nucleobases, the second one is deoxyribose sugar, and third one is a phosphate. There are enormous amount of chemistry involved in the whole structure of the DNA. As we will move on, we will see. So, each of these segments has their particular role, their conformations play a very important role, and so on. I will start with the nucleobases first. These are the 4 nucleobases that are present in DNA: thymine, cytosine, adenine and guanine. Thymine and cytosine, these are present in DNA. RNA has a different one, I will just mention briefly later. Thymine and cytosine, if you look at their structures, they have, both of them has a 6-membered rings, and both of them have this kind of pattern: nitrogen, 2 nitrogen atoms present in 1,3 positions, 1,3 positions. These are called the pyrimidine rings. So, thymine and cytosines are of the nature of, are known as pyrimidine bases, because they have the basic skeleton is pyrimidine structure, which has 6-membered ring, which has 2 nitrogen atoms in 1 and 3 positions. And on the other hand, adenine and guanine have one 6-membered ring fused with a 5-membered ring, 6-membered fused with a 5-membered, and they have of course this skeleton again 1 3 1 3. So, these two are known as purine. They have the basic skeleton that has purine kind of structure. So, these two are known as purine bases. Adenine and guanine are purine bases, thymine and cytosine are pyrimidine bases. This is how the numbering goes. If you look at the thymine, so this NH is 1, 2, 3, 4, 5, 6. This is NH, CO, NH, CO, this is carbon carbon double bond and a methyl group. So, the numbering has to do with, of course, your standard system, that the nitrogen atoms, the heteroatoms should have the numberings which is lowest. In this case is 1, 2, 3. So, you have to start with either you can start here 1 or here 1. So, 1, 2, 3, and the second factor is of course the functional groups has to appear the closest. So, that is why this starts here. So, that the carbonyl will come at the second position. So, 1, 2, 3, 4, 5, 6. So, thymine has basically a methyl group, this is methyl CH3 group at the 5 position, that is important. In RNA, thymine does not exist. What exists is called uracil, has the same structure as thymine, NH, CO, NH, CO double bond and nothing here, H hydrogen, that is uracil, and that is present in RNA. So, in RNA instead of thymine uracil exists. So, this is what thymine looks like. When it comes to cytosine, here you have a amine group, and since there is amine group, there is a double bond and N, it is a difference. So, thymine and cytosine are pyrimide. I will draw the structure of both. It is nitrogen, NH, it is double bond here, and this is a methyl, that is thymine, usually represented by T. Now, when this is connected to the DNA, then of course, it is connected with the sugar, and the sugar comes here in this case here N1 position, numbering is 1, 2, 3, 4, 5, 6 as you have seen. So, sugar is attached to the N1 nitrogen. That means, in nucleoside the NH is not present, only nitrogen is present. So, you have one NH here. Cytosine N, here is your sugar, double bond O, nitrogen, here is an amine. So, therefore, there is a double bond here and nothing, just another double bond. This is cytosine, and together they are known as pyrimide. Now, this is one form. These nucleases, they are actually all aromatic in nature. How? If you look at their resonating structures, and this form can exist in enolate form as well. Here you have a double bond O. So, you can have here N double bond OH methyl. In the free form, if you do not have sugar, then you have the enolate here as well. So, this is the free nucleobase form. This is the enolate structure with 3 double bonds in a 6-membered ring. So, that makes it aromatic. Similarly, the free form here nitrogen in, there will be OH, and this can exist in the other form, that is imine NH. It is basically the nitrogen NHH. That is how the enolate forms is done. This is another form, this is, and when it is re-aromatized, it comes back, then you can have the aromaticity back. So, these are the enolate forms of it, and that makes it aromatic. Similarly, if you look at the thymine of the purine bases, that is adenine and guanine. Adenine is 6-membered ring fused with a 5-membered ring. Here there is one nitrogen and another one here. This is usually attached with a sugar. So, double bond here, so N 1 3, another N, there is amine here. So, that has to be a double bond, and here nothing. So, that is another double bond, that is adenine represented by A. So, if you look at the numbering again the same way, starting with here it will be 1, 2, 3, 4, 5, 6, 7, 8, 9. Number 9, N9 position is attached with the sugar. N sugar, writing S for sugar, then double bond, there is N here. So, it has to be NH, there is another N, there is M in here, that has to be double bond. So, this is guanine represented by G. Now, again as similar to pyrimidine bases, they can also exist in enolate structures and they are also the aromatic compounds. Now, the question is, if I go back, DNA has a specific structure and therefore, the structure or the conformation of the sugar in which form they will be staying in DNA has to be fixed. It cannot move back and forth to enolate as well as the locked form, because in that case the structure of DNA would be flexible. So, question is, out of these two, which one is the correct structure that DNA exists or the nucleobases exists. So, to know that, I will give you the experimental pKa value of the nucleobases. So, if you have a thymine T, T has with sugar in DNA it has a sugar. So, it has free one nitrogen that is accessible. So, that is your N3. So, this is basis and here is the pKa value. The pKa value of N3 of thymine is around 9.93. Now, that means that this NH, this would be deprotonated if the pH of the solution is above 9.93. If the pH is above 9.93, then the NH would be deprotonated and it will exist as N. Below that pH, it will stay as NH. So, now in our body, we call it physiological medium. Physiological pH is around 7.4 actually. So, that is the physiological pH is around 7.4. So, in our body system, we have a pH that is most of the cells, there are other cells also which has low pH. So, 7.4 is the in general the pH of our body. So, in that solution, how the thymine will exist, in which form would exist? It is obviously lower than 9.9. So, NH would stay as this. Therefore, this the enolate form does not exist. This is the form which exists, and that has very important value, because that is very important for base pairing. If you consider enolate form, the base pairing would be completely different compared to the original this form, non-enolate form. Similarly, if you look at cytosine, cytosine also has N3. And that N3 of cytosine has a pKa value which is 4.17. Now, again this means that above 4.17 it will stay as deprotonated form, and physiological pH is 7.4. That means that is already above pH 4.17. So, that means that in cytosine the nitrogen would exist as N, not NH. So, again this is the form that will exist in biological system in DNA, not this form. So, thymine and cytosine both would exist in this form, not the enolate forms. Similarly, if you look at the adenine and guanine, let us consider guanine first. Guanine has one N3 position here as well as N7 here, these two, this is attached to the sugar. So, guanine N3 has a pK, sorry, guanine N1, guanine N1 has a pK of 9.42 and guanine N7, which is this is 7 position, has a pK of 3.3. Now, again using the same idea, you can find out that guanine would exist in under physiological condition, the N1 would exist with the hydrogen attached to it. So, this form, on the other hand, this is 3.3 N7, which means above 3.3 it will be deprotonated and this will stay as deprotonated form. So, this is the correct structure of guanine that will exist in DNA. Similarly, adenine also you can find out adenine N1, I have the value of 3.52. Similarly, adenine N1 is this. So, that will exist in a deprotonated form. So, this would be the proper form that will exist in DNA. So, that is how you see that all the 4 nucleobases that we draw in this way, they are the correct structures that will exist under the physiological conditions. Now comes the orientation of the nucleobase when the sugar is on. If you look at this structure, this is thymine, which is attached with the deoxyribose sugar. Now, it can exist in 2 different conformer. One is, you see here with the double bond of O and NH, the amide bond is in this orientations closer to the oxygen of the sugar. On the other hand, in these structures they are away from the sugar moiety. The double bond, which is planar system, is closer to the sugar moiety here, the double bond here is the away from the sugar. So, these are the 2 conformations that are possible, because this C and N, this is a single bond and therefore, rotation around a single bond is of course feasible under room temperature. So, it can rotate, the bond ideally can rotate. The question is, which is the conformation that will be stable or that will be more favorable. So, this form where this oxygen and this oxygen are closer is called known as the syn form. The other one where they are away is known as the anti form. Now, if you look here, you have a lone pair repulsion, which is absent in this case, and since this is a double bond is planar flat. So, that do not exist any other steric effect also. So, here there is steric interactions as well as lone pair repulsion factor that makes this form, the syn form, less favorable, anti form is more favorable, and this can be clear better when you consider the purine bases. If you consider for example adenine, this is the sugar, this is the N9 of it, N9 of adenine, and then this is the adenine structure, 5-membered ring fused with the 6-membered ring. This is the syn form where the 6-membered ring is just falling on top of the sugar. It is very crowded, very crowded. On the other hand, anti form is nice and smooth with the 6-membered ring away from the sugar moiety. This in the syn form you can understand that there are a lot of lone pair repulsions as well as crowding factors, steric parameters are involved. So, this is not the favorable structures at all, this would be the favorable structure. So, anti form or the anti conformations are the favorable conformations that will be existing in DNA. So, all those factors they actually play vital roles in getting the ultimate specific structure of the DNA. So, what we learn now, we have seen that the structures of the nucleobases are in this form as you usually draw this like this, and the orientation of the nucleobase with the sugar is of the type anti conformations. Now, these 2 factors actually they play role in base pairing. So, they are the ones that gave you the perfect base pairing property. So, this is how the base pairing looks like. Adenine pairs up with thymine, guanine pairs up with cytosines. So, R is the sugar, ribose sugar, that is why I have written R, this is the same sugar. So, now these are the two structures. If you just compare how do you get the base pairings and what is the best possible base pairing to obtain is this. So, before that I will mention one more thing. So, we have seen the thymine exists in this form, the keto form, here there is an N. So, now when you talk about the hydrogen bonding, then there is two factors, one is called the donor, other is the acceptor. Donor represents T, acceptor represents A. Of course, you know all this, I will just give a brief recap. So, if you want to do a hydrogen bond, you need to have a donor which has a donatable hydrogen, H that is donatable. Similarly, the acceptor should have the capacity to accept the hydrogen when it basically leaves kind of like a proton. So, the acceptor should have sufficient electrons to attract the H atom. So, that is what the acceptor is. A typical hydrogen bonding, if you see, usually we represent in D H and then there will be acceptor, then the hydrogen bonding that you get is like this. H is leaving D and coming closer to A, that is what the typical hydrogen bonding is. I will give you one example. So, the like acetic acid, you know, OH, this is where the hydrogen bonding will take place, because this oxygen has lone pair of electrons which can accept the protons. So, this is a acceptor. On the other hand, the acetic acid is an acid. So, this proton is labile, it is acidic proton, it wants to move away. So, that is why this hydrogen bonding will take place, it is very strong hydrogen bonding. So, donor should have a donatable hydrogen and acceptor should have electrons to form the hydrogen bond or to accommodate the proton. So, here are the structures. If you stick to that and the conformations in the anti-formations, then you can see you have a donor here, you have acceptor here, donor here, acceptor here, and for adenine you have a acceptor here and then a donor here. Amine is the donor, here is the nitrogen is free, it has also lone pair of electrons. So, it can accommodate the proton. So, acceptor here, A, donor here, say acceptor, so donor, this is an acceptor. But if you look at the geometry of this sugar, then you can see this CO bond is actually away, it is somewhat in the interior part closer to the sugar. So, that is why it does not participate in the hydrogen bonding. Hydrogen bonding comes from these two factors. Similarly, same thing happens between G and C. Cytosine has 3, this and this. Acceptor, this is acceptor, this is donor. This is acceptor, this is donor, this is donor. Donor acceptor, donor acceptor, acceptor donor. So, 3 hydrogen bonds are possible for GC. If you try to do any other combinations, you will find that this is the best combination possible. So, that is how the Watson-Crick base pairings actually happen. Thank you.